Hi, in this video I am going to talk about bioprospecting of enzymes. Bioprospecting of enzyme can be used to find novel enzyme which has industrial applications. Many industries are using enzymes for the product formation and or as a product. So these enzymes can be isolated from different sources. Maybe we can isolate from animals, maybe from plants, and more importantly from microorganisms. The microorganism is actually the richest source of micro enzymes, because the enzymes produced by the microorganism has many capabilities which is unique. And the one other thing is that microbial enzymes can be easily produced. If you are producing an enzyme from the animal, you need to actually raise the animal to get the enzyme. But if it is a microorganism, it can be easily cultivated in large scale to produce the enzyme. So the microbial enzymes are more important than plant and animal enzymes. So the microbial enzymes can be extracellular or intracellular. So in bioprospecting, we are actually looking for these enzymes to have some novel commercial applications. The screening of enzyme, the process of bioprospecting of enzyme can be of three different types. First one is function-based screening. In this, we are finding the enzyme. We are looking in natural samples to find the enzyme based on the function of the enzyme. If the enzyme is degrading something, we are actually looking at that function of the enzyme to find the enzyme. So the whole idea is to find the enzyme from this nature. This nature has many different number of organisms which is producing maybe thousands of different enzymes. Out of this, we need to find the enzyme which has novel commercial industrial application. So either we can go for the function based screening. In this, we are actually looking to each of the enzyme for its function, or we can do the sequence based screening. In the sequence based screening, we are not actually looking at the microorganism. Instead of that we are actually looking at the DNA sequence of the microorganism. So many people are actually sequencing the DNA and putting it in the databases. So from that database, we are actually looking to the portion of this DNA which may code for an enzyme. The thing is that the enzyme, if you are looking for a hydrolytic enzyme, all the hydrolytic enzymes may have some characteristics, signature sequences. If you can find some type of that signature sequences in a DNA, that portion of DNA may code for a enzyme. So in sequence based screening, we are actually screening the sequences, DNA sequences, to find the enzyme. So the next one is substrate induced gene expression screening. It's a novel method. In this, the genes are screened based on the expression in the presence of a substrate. These enzymes will be, most of the enzymes, extracellular enzymes, will be produced only in the presence of substrate. So the proteins or genes which is getting more expressed during the presence of substrate will be analyzed in substrate induced gene expression screening. So the screening can be done in three different ways: function based screening, sequence based screening, and substrate induced gene expression screening. This screening can be done either in a Petri-plate in a solid medium or in a liquid phase screening. The Petri-plate screening is more easy to do. Then large number of can be visualized. It is a visual screening. Either we can use the colorimetric methods like change in color or something like that. The liquid phase screening is difficult to do when compared to the Petri-plate-based screening. It is done for the soluble enzymes and substrate. It is more quantitative in nature than qualitative. The Petri-plate screening is qualitative. Liquid phase screening is quantitative. And even though the liquid phase screening is difficult to do, its automation is easy. The bioprospecting of enzyme can be done either using screening and selection. In screening, what we are doing, we are actually looking, or we are making a condition in which the particular enzyme producing microorganism will look different. Maybe it will appear in a different color or in a different turbidity. Maybe it is fluorescent or luminescent, something like that will be in the screening. But in selection, we will put a selective pressure in the medium so that the microorganism producing the enzyme or particular enzyme will be the only one growing in the medium. So the bioprospecting can be done either using screening or selection. Screening is easy process, but scaling up is difficult. Selection is difficult to develop, but high throughput means we can do it in huge quantities in automated machines. In this selection, we can either use substrate or substrate analog as a selection force. Think about it. Sometimes we can actually isolate the xenobiotic degrading microorganism in the laboratory. So for that we are using selection process. What we do, we make a medium. In that medium, only available carbon source is the xenobiotic. So, and we will collect soil samples from different places and inoculating into that medium. So this medium has everything except carbon sources. The only available carbon source is the xenobiotic. If any microorganism is growing in that medium, that means this organism is able to degrade the xenobiotic and use that carbon for growth. So that's a type of selection. We'll learn about screening more. Methods of screening. In these sessions, I am going to talk only about screening, not about selection. The particularly useful method is hierarchical screening approach. It has 3 steps because we need to screen for a huge number of microorganisms or huge number of plants. For that, in the first step, what we do, it's a fast and a basic step. It can be done for many organisms. It's qualitative in nature. So in this we are screening only whether the organism is producing some enzymes or not. If it is producing the enzyme, we will go for the secondary screening. So we are doing this primary first level screening for maybe a 1 lakh or 2 lakh samples, and only the samples which show some activity in the first level will go for the second level. Second level is semi-quantitative in nature. We will actually study a bit more about the selected samples in level 2. If they pass the level 2 screening, we'll go for the third screening. Third screening is fully quantitative and is a complete analysis. And this will be done only in the samples which pass through the level 2 of screening. So in this 3-step screening, we'll do the level 1 screening for a huge number of samples, and selected sample will be go to this level 2 screening, and the selected samples from level 2 will go for the level 3. That's why it's called the hierarchical screening. Okay. Next is automated high throughput screening. This is done by automated machines, and it is able to handle 10,000 to 1 lakh samples per week. And it's actually we do using miniaturization and use of microchips. DNA chips is used for finding such duplicates. And we also have the hybrid approaches. In hybrid approach, in the first stage, we will use the hierarchical approach, and the second stage we will use automated machines. So that's what the hierarchical approach is. Then we have more traditional approaches like chromogenic substrate screening. So one such example is Indigogenic substrate. The enzyme beta-galactosidase, you might have heard about it. It's a part of lactobacillus. If you want to find the microorganism which is producing beta-galactosidase, what we do, we'll make a medium which contain a chemical called xgal. Actually, the real name of enzyme is 5-bromo-4-chloro-3-indolyl galactopyranoside, but it's abbreviated as xgal. The medium is containing xgal. Xgal is a colorless compound, and you are taking microorganisms from different sources and just plating in the Petri-plate, just like you are doing in the laboratory. If any of this microorganism is producing beta-galactosidase, what it will do? It will degrade this xgal to produce a blue colored compound. So if any of this microorganism is producing beta-galactosidase, the colonies look blue. So you can actually identify the organisms which is producing beta-galactosidase. So that's in that way we can actually isolate the microorganism which is able to produce beta-galactosidase, and which we can actually industrially use this microorganism for commercial production, if any. Same thing can be done for amylase production. It's a classical method. Amylase is the enzyme which degrades starch into simple sugars. And the starch has a reaction with the iodine. Like if you add iodine into a starch solution, the solution will become dark blue. So the reaction of starch and iodine is very well known. So what we do here is that we will make an agar. It's called a starch agar. The starch agar, the carbon source is starch, just like our agar, nutrient agar, but the carbon source is starch, and it has a lot of starch in it. We will make a Petri plate of starch agar, and we'll take samples from different places and plate it over the starch agar. And after incubation, what we'll do is like the microorganism which is able to produce amylase will produce amylase and degrade starch present in the medium. So starch will be degraded into glucose, and this microorganism utilize the starch to grow. So around the colonies of starch amylase producing organism, there will be degradation of starch. There won't be any starch. After incubation, what we do, we will add iodine into the medium. So if we add iodine to the medium, iodine will go and bind to the starch and produce a blue color. But any microorganism which already degraded the starch, that area won't be having the starch, so iodine cannot go and bind there. It won't produce blue color. Just like this figure, this colony, this microorganism is not producing amylase, so the blue color is visible everywhere. But colony here, it was small, just like this, is actually producing the enzyme amylase and degrading starch around it. So when we add iodine around this colony, we are seeing a clearing zone, not blue in color. So that's way we can actually identify the organism which is producing amylase. Fluorogenic substrate can be used in screening, which has very interesting applications and it is very sensitive in nature. One such example is the cow marins. These cow marins can be used for the screening of cytochrome P53, P40, cytochrome P450. It's called CYP. So here we have two cow marin derivatives. These cow marin derivatives are non-fluorescent in nature. But any microorganism is producing this CYP. The CYP will conjugate these two or modify this thing to produce fluorescent compounds. So this activity of enzyme is converting the non-fluorescent compound into a fluorescent compound. So in the plate, if you are seeing some fluorescence, that means the organism is producing CYP. So that way we can actually identify. Finally, we have the metagenomics. Metagenomics is actually the analysis of genetic component of the entire habitat. You might have heard about the eDNA. eDNA is called the environmental DNA. In the DNA sequencing approaches, traditionally what we have done, we will take the organism, isolate the DNA and go for the sequencing. But in metagenomics, we will take the environmental sample because most of these environmental samples are non-culturable microorganism, the microorganism which cannot be cultured in the laboratory. So we'll take the sample from a habitat and sequence all possible DNA within that sample. And from that DNA, the sequence will be used for the further processing. Or this DNA is used to make clones. And these clones will be identified for the finding novel enzymes. So here is a schematic representation. We are taking the samples. We are taking the samples and isolating DNA. This DNA is cloned into the vectors, and these vectors are cloned into the host organism like Escherichia coli. And we are actually screening this Escherichia coli for the novel enzyme. Okay, the original organism which is actually producing the enzyme may not grow in the laboratory. So we are taking DNA and putting it in a host organism, and we are doing the screening. That type of screening is called metagenomic screening. Here is the whole step-by process: extraction of DNA from microbial niche. Then we are producing a DNA library. DNA library means these DNAs are cloned into Escherichia coli, and these clones of DNA can be either long insert or short insert. You don't have to worry about it. And for the long insert, what we do, we do the function based screening. So whether the colonies which is harboring the DNA is able to produce an effect. For the short insert, we will do a sequence based analysis, and once we found an interesting application, we will take the DNA and go for further studies. Thank you so much for listening.